So today I'm going to be talking about somatic cell nuclear transfer, and just some basics about the technique would be that the definition would be a technique for cloning. This technique was founded in 1996 by Ian, Wilma, and Keith Campbell with their reproductive clone of Dolly the lamb, and I'll go more into their experiment later on, but first I want to talk about the physical process of somatic cell nuclear transfer. So there are two different types of somatic cell nuclear transfer, including therapeutic somatic cell nuclear transfer and for the use of reproductive cloning. For the sake of this research paper, I focused on reproductive cloning. So in that, there are 5 different steps that you can take to achieve a clone. The first being that you would remove the nucleus from a healthy egg, and then you would extract the nucleus from a donor cell, such as the skin, hair, muscle cell, etc. The third step would be that you would insert the donor nucleus into the healthy egg, and then the zygote would undergo the phases of meiosis, and you would have your clonal embryo. In my next slide here, I just have an image that kind of explains or visually interprets that process. So on the left-hand side, you can see that you have a female egg, and then you have a body cell of the donor cell, in which both of those, the nucleus, are removed. And then you would take the nucleus from the donor cell and insert it into the egg, and then you would have your clonal zygote, which would then be your clonal embryo. So now that you understand the process of somatic cell nuclear transfer, we can go back to Ian, Wilma, and Keith Campbell's experiment of 1996. So to achieve their first adult clone of Dolly the lamb, they derived adult sheep mammary cells. And ultimately their success was determined by Dolly's survival. So Dolly the lamb lived to be about 20 years old. She reproduced 6 little lambs, and she ultimately died because of a tumor in her lungs with no association to the fact that she was a cloned animal. As for impacts on human society, I first want to talk about positive impacts. So the positives would be that you can clone animals for various purposes, which I will go into more on the next slide. You can clone your pets for personal reasons, and it has this technique has the potential for curing mitochondrial diseases, mitochondrial-related diseases. As for cloning of animals, there are 2 main positive impacts. For agricultural purposes, you can have mass production of domesticated livestock in developed countries where there is a lack thereof. For endangered species, we can bring back populations and help ecosystems that are in need. As for negative impacts on human society, the clones are not 100% matches. In some of the research that they have done, the clones don't match exactly in their personalities. The clones also have a high fetal death rate. And along with that, the cost of somatic cell nuclear transfer is extremely expensive. Just to reproduce Dolly, the cost was $750,000. And according to Viagen Pets, you can clone your personal pet for a dog would be $50,000 and a cat would be $25,000. So in conclusion, the somatic cell nuclear transfer technique is a perfect example of different fields crossing over, such as technology and genetics. The technique shows promise in positive impacts of animal cloning, and it is yet to be discovered or really experimented with, but there is the potential for the human use of somatic cell nuclear transfer. Thank you.